my name is Xiaoqi, and then today I'm going to talk about how tree growth and how tree growth and the community dynamics of tropical forest respond to climate change. So here's some background information and several questions that we are interested to answer. So basically, tropical forests, they're a very important carbon sink. They're responsible for like around 25% of the terrestrial carbon sequestration. And climate change is predicted to affect this ecosystem. But how tropical forests respond to climate change is very, very difficult to predict because the extreme high species diversity in this ecosystem and the complicated links between tree physiology and tree demography. So here we have asked two major questions. So the first one is how shifts in this climatic conditions going to affect the tree growth in tropical region, and then the second one is how the physiology of these trees affect the effects of climate on tree growth. And here's our study site. Our study site is in the El Yunque forest in Puerto Rico, located in the east-north corner of Puerto Rico. We have several plots established in this forest. First one is the Luquillo Forest Dynamics plot. This plot is relatively big. It's a 16-hectare plot. And then we have another 4 chronosequence plots established around these big plots. So the sizes of these plots are relatively small. They range from half hectare to 1 hectare. Luckily, this project started with a lot of data. So our co-researchers have already started to collect data since 5 years ago. So for the first big plot, we have dendrometer bands installed on the 900 trees in this plot. So the dendrometer bands are just used to measure the diameter growth of these trees. And then these measurements were collected from 2012 to 2015, and then we're going to continue it in 2016. And then here's just some simple analysis of this data set. So so you can see that these are the diameters of the trees. You can see that from 2012 to 2015, the diameters are actually getting bigger and bigger. That means the trees are growing, so which is normal. And then if you use current year's diameter subtract last year's diameter, then you get the growth of the tree, and then which is shown in the next slide. So for the first 2 periods, so 2012 to 2014, there were no difference among the growth. And then in the last 2 years, from 2014 to 2015, you can see there's a significant drop of growth in the tree, which is coincidentally happening with a drought that happened in the growing season of 2015. I will show you the rainfall data later. And then so this is our first data set, and then our second data set is the chronosequence plots. So there are 4 plots over here: EVI, SB1, SB2, and SB3. So the EVI, that plot is established next to the El Verda station, and then the other 3 plots were set up next to the Sabana field station. So you can see which is, here it is. So here's the El Verda station over here, and then the other one is the Savannah Field Research Station. And then these plots are in different successional stages, and then we collected the DBH, the tree growth data from 2011 to 2015. And then here's some simple results of this data set. So same here. There's a growth reduction over here from 2014 to 2015. So this is the absolute growth, and then if we normalize the absolute growth by the tree size, which we get the relative growth rate, you can see the same pattern over here. So the growth rate from 2012 to 2014 around 2% 1 year, and then but the annual growth reduced to like around 1% from 2014 to 2015, which is also probably caused by the drought. However, we don't see a increase in mortality rate of the trees. We expect to see an increase in the mortality rate of the trees due to the drought, but we didn't see anything yet, probably due to the lag effect. Probably we will see something in next year's data. This is just, we added another year's data from 2011 to 2012. But we still didn't see any patterns over here. Initially, the 2011 to 2012, that year's data was relatively noisy, so we decided to not include it in any other analysis. So here the rainfall data, you can see this is from 2010 January to 2015 November. You can see that in the growing season of 2015, there's a severe drought that has happened, which may have caused the reduction in growth that I have mentioned earlier. And then, so we did analysis on the reduction of DBH during the drought period, and then we categorized the trees by small trees and large trees. Small trees has a DBH of less than 10 centimeter, and the large trees has a DBH larger than 10 centimeters. So you can see that during the drought period, for both small trees and the large trees, the DBH growth has reduced. But then when we normalize this reduction by tree size, you can see that actually small trees suffer more than large trees during the drought period. The reduction of growth rate was almost as high as 4 to 5 times of the reduction in large trees. So so based on this simple analysis, we established a hierarchical Bayesian model to model the tree growth using this biotic factor, which is the DBH, and then the abiotic factor, which are the climate conditions. So DBH is the diameter of the tree, and then NCI is the neighborhood crowding, which is the index of how much light this tree get, and then the VPD is vapor pressure deficiency, which is an index of water stress of this tree. So So we're using this covariance to model the growth of the trees in each year. So if you're not familiar with this professional terms, you don't worry about it. So Bayesian is just an alternative way to do statistics than Fisher statistics. And then hierarchical means we have more than one level of parameters in our model. So this is the first level, and then in the second level, we also modeled how tree physiology, basically specific leaf area and wood density, affects each parameters in these models. So here's our modeling results. So this is a figure that's showing predicted growth versus observed growth, because our model restrict that the growth has to be positive. So you don't see any negative predicted growth over here, but due to human error, we have like a good portion of negative growth in the observed data set. And then this figure shows you how SLA and wood density modifies the effects of climate change on tree growth. I don't want to go through the detailed math over here. I will just use the BPD as an example, which is the index of water stress. So a negative value over here means higher SLA will intensify the water stress, the negative water stress on tree growth. So basically, if you have a higher SLA, that means you will have a higher reduction of growth during water stress. And then, so over here, wood density. So for this value, so if you have a higher wood density, then your reduction of growth during drought will be smaller. Okay. And then, so when we plot this data out, you can see that when SLA goes higher, and then during the drought period, the growth rate increases. Actually, this makes perfect sense because SLA means the amount of leaf area a tree has over the amount of mass. So if you have a higher area, then you will have higher water evaporation. So that is definitely not good during drought. So that's why if you have a SLA, higher SLA, then the reduction of the growth during drought will be higher because your water evaporation is much higher. And then over here, this slide shows you if you have denser wood, then if a drought happens, then the reduction of the growth rate is less. So to summarize, these are my findings. So drought reduces growth rate for both small trees and large trees, but small trees suffers a lot more than big trees. And then tree species with higher wood density suffer lower growth reduction during word stress. And the high SLA has magnified the negative effects of drought on tree growth. If we have a sustained drought, then the low SLA species probably will tend to dominate in late successional tropical forests. And then we didn't see anything in mortality rate yet, but we hope that we can see something like with further data collection. And then finally, I want to thank my supervisor, Maria Briati, for providing this data and for her suggestions. And then for my technician, Laura, for her help in coding and programming. I want to thank everyone in the Briati lab for their help. And I want to thank Earth Institute and the committees for sponsoring me to work in this position and this project, and specifically Gavi for her help in many matters for my transition to this position. Thank you.
Thank you, very interesting. Other than the water effect, can you detect, and is there a way in this research designed to detect direct effects of temperature, for example, because of interannual temperature variability? And also, is there any way, even though all trees are sharing the same carbon environment, to look at changing carbon concentration and carbon fertilization for the trees?
Potentially we can. So in the model that we're using over here, So this model, so these are the direct effect of DBH, light and water stress on growth. I didn't show the values over here in this presentation. I was afraid that the presentation would be so too long. So basically, actually I wanted to add another parameter, which is the temperature, minimum temperature in the night, so I can also see the warming effect on this, the tree growth. But I have done a similar work before. The minimum temperature, actually the effect was not significant, so I started with a simpler model at the beginning. And then for the carbon sinks, I am doing another project which is using an ecosystem model to model carbon sinks, like in the next 50 years and 100 years in the tropical ecosystem. But the difficulty things is model validation. I mean, we can make as many predictions as we can, but the thing is that we have to have enough data to validate the model to make sure that our model is performing properly. So right now we only have plot data from this site. We don't have any covariance data or anything from this site. That's the biggest problem right now. I'm trying to figure out to collect as many data as possible to make sure that my model is correct.
Very good. Thank you.
Thank you. Do you have or do you plan to gather any data on soil chemistry?
We have soil chemistry from the site. So we have soil carbon, soil nitrogen, and soil moisture, and some seedling data from this site. So there are a lot of data that's available. It's just how we decide to use this data.
In another study that I'm working on, actually, soil calcium seems to be a major player in tree growth. Do you have any information about soil calcium?
I'm not familiar with this set.
Okay. It would be interesting to see whether any of your chemical variables influence the sites, are of influence for the site-specific data.
Okay, cool. Yeah, I need to check that up. I haven't looked through the soil chemistry data like really clearly. I mean, really close yet. So it's nice to know that. Thank you.